Hey guys, welcome back to the channel. So in today's video, we will be talking about the excitation contraction coupling. We will be talking about how myosin heads perform the power strokes and how sliding filament model works and how that all get to happen. And we'll be talking about also like muscle tension and the factors affecting it, the different types of muscle fibers, muscle type. So we've got fast twitch and slow twitch, right? And we'll also be talking about length tension relationship. So let's first start off with what we call the what happens at the T-tubules, right? So in the previous video, we did cover the fact that T-tubules are extensions or invaginations of the surface membrane of the muscle fiber called the sarcolemma. So this is the sarcolemma, right? And this is T-tubule right here. So these are extensions that go deep into the muscle fiber. So the sarcolemma is superficial and the T-tubules go deep into, and their main function is to conduct action potential, which travels down and reaches the myofibrils inside better. Right, so at the tubule membrane, you've got this protein right here called the DHPR, voltage gated protein channel. Right. And over here, you've got this, what we call this is the sarcoplasmic reticulum, basically. And you've got this, right here, this is the RYR, or the, that's the name for it, but the actual, this is an abbreviation. The actual name is ryadenine receptor. So it's ryanodine receptor. Right. We've also got on the membrane, we've got another protein pump here. We call this the SERCA. Right. Again, it's an abbreviation. It stands for sarco endoplasmic reticulum. Sarcoendoplasmic reticulum calcium ATPase. All right, and I'll go through the function of each one of them in a moment. All right, so what happens is as the action potential reaches, so we've got the motoneuron here, the presynaptic motoneuron here. It sends out ACH molecules, right, and then a bunch of things happened, which I covered in the previous video. I won't be going again. That generates a new action potential in the muscle fiber, and the action potential travels along the sarcolemma and down the tubules to reach the basically the sarcoplasmic reticulum. So this entire bit right here is what we call the terminal cisternae. So this entire thing is what we call the terminal cisternae of the sarcoplasmic reticulum. All right. And what happens is that, imagine you've got this, right, the T-tubule here, you've got one of the terminal cisternae here, and another terminal cisternae facing the other way, like this. These three together would make a triad, what we call a triad, right? And again, I've covered that in the previous video, so please go check it out. Now, what happens is that as the action potential travels down the T-tubules, it will activate the voltage-gated DHPR protein channels, right? And it will allow calcium ions to flow through. So as calcium ions flow through, the RYR, the ryanodine receptor, detects the calcium, the presence of calcium ions, right? And the receptor will open up and letting out even more calcium ions from the terminal cisternae of the sarcoplasmic reticulum, right? So as we know, sarcoplasmic reticulum's main function is to store calcium ions, right? And as the ranitidine receptor detects the presence of calcium ions outside in the sarcoplasm, it will open up and allow calcium ions from inside the sarcoplasmic reticulum to flow out through it and into the space around. And what happens is it will travel down to the myofibrils, where we have, of course, the sarcomere, the functional unit of contraction. This is what it looks like. All right, so this is basically a sarcomere. This entire thing is called the myofibril. One of this guy is called the sarcomere. Right, and again, I've covered this in the previous video, but essentially the calcium ions will bind onto a protein called the troponin on the actin, and allowing the myosin heads to be able to bind to the actin and perform power strokes and, contract or flex or contract. Sliding monofilament, what happens, like where the myosin in the middle and the two actins come together towards the M line, or the two Z lines come together closer. Right, that's the sliding monofilament. And so calcium ions allow that to happen. Right, so after stimulation has ended, or the stimuli has stopped, or your muscle could be experiencing fatigue, then this is when the circuit comes into place. So the circuit, which is basically an active pump, will pump calcium ions back inside the sarcoplasmic reticulum for recycling purposes, because you don't want to waste all those ions, and we can reuse them when your muscles need to contract again. And we use ATP in this process to pump calcium ions back in. Right, and this is essentially the cycle of excitation, contraction, coupling, reaction. Right. So what really happens in this cycle? Because now I'll be going over how all those contraction really happens. So what happens is that you've got this guy here. This is the actin. You've got, let's say, these two binding sites up here, and you've got a myosin head. Right, so go on just like that. So what happens is ATP is attached to the myosin head, and the myosin head will bind to the actin like this. Right, ATP, just like that. And it will perform a power stroke. So we'll pull it backwards. So now the myosin is positioned like this. Oh, just give me a second. Yep, so what happens next is you've got this gap over here, and now your actin has moved forward slightly, and now ATP will basically hydrolyze to form ADP, PI, inorganic phosphate, allowing the myosin head to detach from the first binding site. Right, next is the myosin head will cock backwards, will reach out, and basically bind like this. Right, ADP, sorry, I'll just redraw this diagram of here. Right, so I'll just go through from the start again. that ATP. So basically, for the first step, right, you've got your myosin head attaching to the first myosin binding site on the actin. Second step is that the inorganic phosphate will leave the myosin head, changing its conformation and allowing it to flex and pull backwards. The third step is that ATP will hydrolyze, then become ADP and inorganic phosphate, allowing the myosin head to detach from the binding site. The fourth step is that the head will cock backwards and again bind to the next binding site along, right? So here you've got, it's binded to this one. Now here it's binded to the second one, right? So it shows the actin has moved forward while the myosin head stays still and it only cocks backwards or changed its angle, basically. And again, we are now back to the first cycle. So this is basically muscle contraction cycle, right? And so that is basically it, and we'll move on to muscle fiber types, right? So you've got fast twitch and slow twitch. So this is all you need to know for fast versus slow, is that I'll construct a table which will make it easier for you guys. Right, so the first heading is the diameter of the fiber, and of course fast fiber larger, these guys are smaller. Then you've got capillary supply, or if it's vascular or not. Fast is less vascular, these guys are higher, or higher/more vascular. Right, mitochondria supply. Fast has less, slow has more. Fatigue resistance. This guy is little. This guy's a lot. Time to peak tension. Time to peak tension. Fast, slow. That's why these are called fast twitch and slow twitch. And color, these guys are white and these guys are red, basically. So these are the main comparisons between the two types of fibers. And so slow twitch fibers are designed to last for a long time. When you've reached tetanus, when your muscles are in contraction for a prolonged period of time, and you're not just suddenly twitching your body and you're holding it still. And fast is for when you need that quick twitch or quick tension from the muscles. Like, for example, if you're accelerating, if you're sprinting, you need a quick boost when running, right? So that's basically different types of fibers, and the colors as well. You have to know the colors on the slides when presented to you during class. Now we'll talk about length tension relationship, right? So we all know that muscles contract, but they also have an optimal length, right? What I mean by that is that these guys, right, for example, if I show you 3 diagrams actually. Boom, boom. Third diagram is this. All right, so the difference is that here, the two Z lines are really far away. Here they're moderately far away, and here they're really close together. All right, so what happens is that this is assumed to be optimal. This guy, there's really no overlapping between myosin and actin, so there's really no force that this muscle can do. So these are overextended, right? Or if they were to contract, the force will be really weak. This guy will be relatively strong. It will be actually the strongest, basically, because it's had just that perfect amount of contact for enough myosin heads to bind to and contract, but also leave enough space for the Z lines to come together, right? Here, the thin filaments are too close together, So they can't imagine if you're crossing your hand like this. If they're really far apart, then they're not even touching each other, or touching a little bit, then they'll go very weak. If they're middle like this, in the middle, then the force of contractions are very strong. And if they're really close like this, then they can't really contract anymore, and the force or the tension produced will be weak. That's why we have this optimal length there, when the tension or the force produced will be at its peak. And this works for every single muscles as well. So depending on what muscles they are, they will have a different length tension relationship. We also need to know about muscle tension as well, and I'll draw this graph over here. So what happens here, imagine this. All right, and I'll draw this. So what happens is that this is the action potential, this y-axis, and this y-axis is contractile activity / force. So basically, when an action potential is sensed, right, as you can see, I know my drawing may not be the best, but its intensity, its amplitude is the same amplitude, but different frequency. But here you've got increasing frequency as you go along, right? So what happens is, if you've got a single action potential, your muscles will twitch like that, right? Hence it's like this. No, but if you increase the frequency of the action potential, they may add up, right? So this is twitch summation. What happens is that you're adding up your twitch together to form a more a stronger tension of force, right? But if you have very frequent action potentials, these twitch will add up really large until you've reached maximum contractile force, and this is called tetanus. Tetanus, tetanus. This is when your muscles are basically, you contract them up and you keep them contracted, right? And this is what tetanus is, when your brain keeps sending signals to your muscles saying that you have to keep contracting it. So action potential frequency is really high now. And this is basically muscle tension. And muscle tension is also based on, first of all, the number of neurons activated, right? So, and therefore the number of fibers are recruited, right? So, a motor unit is composed of a motor neuron and all the different muscle fibers that it controls or it innervates, right? So ultimately muscle tension depends on the number of muscle fibers recruited or activated by the neurons, and also depends on the stimulation frequency here, or the action potential frequency. So those are the two main things that affects muscle tension. Yep. And also tetanus is defined as maximal prolonged contraction of muscles due to repeated stimuli. Thank you for watching today's video, and I'll see you guys in the next one.